Morning, everyone. Just let me know if you can see the video feed properly, because the YouTube one's for some reason saying it's private, but it should be streaming okay. So just let me know if the video's coming through. Hope everyone's well. This is part 4, possibly the final part of Biology 3. So we shall see how it goes. Morning, Tanishka. Morning, Chris. Not sure who the third person is. Don't recognise the name you've called yourself. So let me know. So we've looked at so far... brilliant. Thanks for that, Chris. So we've looked so far at pathogens, what they are, microorganisms, different types of microorganisms. And we've looked at some of the diseases that you need to know about. Remember, we said that actually they can ask you about different diseases, but they just need to make sure that... Brilliant. Morning, Jack. Cool. Morning, Nico. We just need to make sure that you can ask questions about other ones as well if they give you enough information about it. So we've also looked at... actually, what have we looked at? I think last time we were going through... Bring up my notes. That's right, we looked at drug development, we looked at preclinical trials, we looked at clinical trials, and we looked at actually how it's a really long process in order to be able to develop new drugs and bring them to market. So we went through and we sort of looked at what that's involved with. We also looked at MRSA and resistant strains of things like gonorrhea, so where they're resistant to penicillin. And we linked it into evolution. And we said that actually we only need one mutation and one bacteria cell to become resistant, and all the others get killed off by the antibiotic. And that one can then multiply and we've got a new resistant strain. So we looked at some of the techniques as well about avoiding more strains becoming resistant. So make sure that you go back through some of that. Now, what I want to have a look at today then is we're going to have a look at something called monoclonal antibodies. Bring up my notes. Monoclonal antibodies. We're going to link it back to the antibodies and antigens, because actually I suppose monoclonal antibodies really proves whether you do or don't understand about antibodies and antigens. So we'll use it as a recap on what they are, why they're useful. We'll then talk about how monoclonal antibodies are produced and some of the uses for them, because actually it's quite relevant, especially now, and it's still technology that we're developing into further areas, everything from cancer treatment through to testing whether you've had a particular disease or not. So we can have a look at that as well. And some of the testing for COVID-19 will be based on monoclonal antibody tests. So we're going to have a look at that as well. Now, depending on how long that takes and how much I waffle on and we go through and we look at that one, and obviously if you guys have any questions as well, we may cover plant disease today as well. If not, we'll probably just do the culturing of microorganisms and do plant disease next time round. I will run a Google Meet straight afterwards. So as usual, I'm going to put the class code or the Meet code for that into Google Classroom. So if you about 2 or 3 minutes after the video ends, so keep an eye on there, refresh the page after about 5 minutes, and you should see a link appear. Right, let's make a proper start. So we've looked at this idea then of antibodies and antigens. So we're going to start, and I'll just put a title in here. There we go, monoclonal, let's be lazy, antibodies. Now, remember we looked at different types of white blood cells. And we said we've got the white blood cells called lymphocytes. And we've got the white blood cells called phagocytes. There are other ones as well, but those are the main ones that you need to know for GCSE. We said that lymphocytes produce antitoxins. They also produce antibodies. So their job is to try and recognise invaders. They're trying to recognise different protein structures on the surface of a foreign invader or a foreign body or a different type of molecule. And if it recognises that molecule shouldn't exist in the body, it's going to produce antibodies. So antibodies bind themselves to antigens. And this is really key to their use, because what it means, what it means is we can actually tag different foreign substances in the body, and we can either do things with it. So we can either have it flagging up on a test kit as it's now present, so we know it's there. So for instance, if we knew the antigens on the surface of COVID-19, you could have a blood sample tested. We could then run that test in a small test kit, and if it changed colour, then it could indicate that you had COVID-19. So we're looking at tests like that. Now, whether they've got to that stage yet, I don't know, but I know it is used for the treatment of recognition of some other diseases, and we'll look at how that works in a minute. We can also use it in pregnancy test kits, and that's one of the sort of situations and scenarios that we're going to have a look at today. We can use it for testing for cancer, or treatment in some cases for cancer, and they're looking at it now as well. It's quite a new research area, but it is an area that's going to grow. So it's quite important to try and get our heads around what's going on with it. We'll also have a look at how there are some disadvantages of monoclonal antibodies in the way that we're doing it at the moment. So we'll have a look at that as well. But the key thing for all of this is this idea that antibodies binds to, and if I want to be really sort of pedantic, I'm going to put specific in here as well. So in other words, each antibody is unique to a specific antigen, in the same way that enzymes are specific to their substrate. So let's look at the process for production of this. I'm going to stick with blue for a minute. So we've got production of, this is going to be our monoclonal antibodies. I'm just going to put that down for short, okay, or even just MABs. So what we do is we actually inject mice with the antigens. Now this could be this could be a protein, it could be an enzyme, it could be whatever contains the antigens that we want the antibodies for. So we're going to inject mice into that. Then the next stage what we need to do is we need to extract the antibodies as they're produced. So if you think about what's happening here, we're injecting the mice with the antigens. Their lymphocytes in the mice are going to produce antibodies. Okay, in the same way that when you get infected with a new disease, we feel ill for a little while if it's the first time you've had that disease, and that's because your body has to learn to recognise that you have something new in your body that shouldn't be there. Your lymphocytes then will recognise the antigen, so these particular protein structures, and it will start producing antibodies that fit or match those antigens. And that's what's happening in the mice. We're going to deliberately inject the mice with antigens. We're going to leave them in there for a little while, and the mice's immune system, the lymphocytes in the immune system, are going to start producing antibodies to try and get rid of those antigens. Okay, and if you remember what we said before, that in, I think it was video 2, so if you're not sure, go back and have a quick look, but the antibodies, when they bind, sometimes they cause the pathogen to break apart, but they can also cause them to clump together so they can be recognised and destroyed by the phagocytes, the other type of white blood cells that you need to know about. So what we're going to do is we're going to leave the mice for a while, and we're going to let those antibodies be produced by the lymphocytes. So the next job that we want to do, and I've just made a mistake here actually to get rid of that. Let's get rid of that bit there. The next thing we want to do is we actually need to extract the lymphocytes. Okay, let's just get rid of that. So we need to extract the lymphocytes from the mice. Now, this can be done, generally, it's often done by looking at the spleen. So it's often produced from the spleen. Sometimes it can be produced from the blood of the mice. Often, this leads to death of the mouse. Okay, so that would be one disadvantage for some people when we're looking at this one. So I'm just going to put that down there. It often leads to the death of the mice. So for some people, that would be a disadvantage of using monoclonal antibodies. So the next step then is we need lots of these. So we need to produce we need to increase the number. So we need a way of producing lots of them. So what they do is the next step is they combine the lymphocyte Remember, the lymphocyte is just another name for a type of white blood cell, part of your immune system. So they combine it with a cancer cell. Okay? This won't be done inside the body. This will be done in the lab. And this produces a hybridoma. Hybrid because we're looking at this combination of two cells. Now, it's quite important that we understand why we've linked it to a cancer cell. So, cancer cells multiply very, very fast. And that's one of the reasons they cause issues inside the body. In a lot of cases, a cancer cell is very similar to one of your body's normal cells because it would have been produced from one of your body's normal cells. And that's why your own immune system doesn't normally attack it, which is why we don't get rid of it and we need other types of treatment. Basically what's happened is there's been a mutation in one of your cells and it causes those cells to start multiplying much too fast, faster than anything else around them, which is why we end up with some form of growth. Now, when we're looking at the different types of cancer, and we'll talk about this later on when we get to a different topic, but when we look at different types of cancer, we either have benign, which is where the growth stays localized, or we have malignant, where they can spread around the body. Now, in this case, this isn't being done inside an organism, so the cancer cell itself isn't at risk. But basically what they've done is they've taken a cancer cell, they've combined the lymphocyte with that cancer cell, and then they're allowing it to multiply very, very fast. Because what this is going to do is produce large numbers of lymphocytes, and all of those lymphocytes are going to be producing antibodies. So we're going to allow to multiply. And again, I've just changed colour so we can put the reasons down here. So we're going to get large numbers of lymphocytes. Oh, can't spell today. There we go. And that's going to equal large numbers of antibodies. Okay? And remember, it's the antibodies that we're after here. All of this is to try and produce these antibodies that we can do things with. And those things we're going to look at in a minute. So, make sure you can understand this one. And I'll just put the last bit down. We're then going to purify the antibodies. And what that means is we're going to collect the antibodies, we're going to get rid of all the other cells, everything else that we don't need, so we're just left with literally lots and lots of antibodies that we can then do useful things with. So let me just run back through that very quickly, just to make sure that you've got your head around how we produce these. It's worth noting when we're looking at this though, for a minute, let's just break down where these words come from. So we know that mono means one. So we're looking at one type. We've got this word clone in here. So this is going to be the same. Remember, if you clone something, you're making lots of them. So we've got one type or the same. So cloning, actually, let's just put that up here. Production of the same. So we've got one type and then we've got our antibodies, which we obviously know about. So there's three parts to the word. So everything we're doing is looking at large-scale production of one type of antibody. Okay? So we inject the mice with the antigens. The mice's immune system goes, those antigens don't belong in me. So it activates the white blood cells. The lymphocytes then recognize the antigens and they learn to produce an antibody that is designed to fit that antigen. So if the antigen had a shape here, let's just say it had a shape like that, then we may be getting some antibodies produced with the shape that's going to fit. Okay, it's more complicated than that, but as far as we're concerned, that's what we need to know. We've got the antigen and then the lymphocytes produce the antibodies which are designed to fit it. Once we've got those lymphocytes that know how to produce the antibodies, we're going to extract those lymphocytes, often from the spleen, but usually causing the death of the mice, so it's a disadvantage. We want to increase the number of them so we can produce large scale, and we're looking at industrial scale numbers of these antibodies. So to do that, we combine the lymphocyte with a cancer cell, and then that cancer cell produces a hybridoma. That's just the name of the combined cell. So don't worry about it being anything technical. It's just the name of those combined cells. We allow the hybridoma to multiply. So that's here. And then that means as it's multiplying, we've got large numbers of lymphocytes, and we can then just sit back and wait for those lymphocytes to start producing huge amounts of antibodies. And as long as we have the correct nutrients, the cells keep multiplying, the number of antibodies keep multiplying, all we have to do is extract and purify the antibodies so that we can then go and use them in whichever industrial process we need to. Okay? So make sure that's fairly clear. It's basically what happens inside our body. We're just scaling it up so we can actually produce it on an industrial level. Like I said, if you think about the number of pregnancy kits that use, pretty much all pregnancy kits will use monoclonal antibodies, and we're going to look exactly how they do that in a moment. Still keeping my coffee well away from my pen at the moment after dropping it in there the other day. So let's look at the first use for this then. And this use is going to be in pregnancy tests. So what we have, and you're going to apologise my drawing, it's going to be my usual technical, but in no way, in any way, shape or form, artistic drawing. This is going to be our pregnancy test kit, and I'm going to do it as if you're looking sideways on, because that way I can annotate different diagrams on there. There are lots of diagrams on YouTube and on Google for this one, so I'll show you some other diagrams in a minute that you can have a look at if my one isn't making too much sense. Now, what we have here is, let's go for a different colour. We're going to have a reaction site. Oh, go back to pen. We're going to have a reaction site here. The reaction site literally is just the area that gets peed on for the pregnancy test. So the female is going to pee on this particular area. Now we call it a reaction site because here we're going to have some different antibodies. Sorry, not different, we're going to have some monoclonal antibodies. Okay, so we're going to have our MABs, monoclonal antibodies. Now these are going to be specific antibodies for a hormone called HCG. Okay, so we're going to get, urine's going to go here, and I'll put a note on here, if pregnant, it contains HCG, and that's, HCH, what am I doing? HCG, there we go, so that's just the name of the hormone that you need to know. You don't need to know what it stands for. You can go and look it up. That's absolutely fine. There's loads of things online. Just remember those letters. And if you can't remember the letters, just remember that it's a hormone that's present during pregnancy. Okay? Remember that monoclonal antibodies is quite high up when we're looking at sort of complexity. So try and simplify it as much as you can in your head. And if you've got specific questions while we're going through this, write them down. You can put them in the comments box, or write them down on a bit of paper and ask me in Google Classroom afterwards, because I'm more than happy to go through some of this when we do our Q&A session later on. So we'll carry on going through. So let's just say, for instance, and I'm going to make this really simple, that our HCG hormone is a round ball. Okay, because it makes the next bit much simpler when I'm drawing the hormones. So we'll call this our HCG hormone. Okay. Now, what's going to happen is if HCG is present, we're going to get the antibodies Okay, so these are our antibodies. our monoclonal antibodies that's been produced, because we only want one type of antibody on there. So if the HCG is present, what we're going to find is they're going to bind themselves to our antibodies. Okay, so that's all that's happening. When we're talking about a reaction site, all that's happening is the hormone itself, if it's present, binds itself into that antibody. Now, the antibody is going to have, it normally has some form of dye on there. It won't change colour yet. So this is a particular type of dye. Now what happens is the urine moves down here. And we end up with the test site. Okay? So the urine's going to gradually work its way up. A bit like, if you remember when you've done chromatography in school, where we had the filter paper, and what we did is we put some pen on the filter paper, we put it in a little bit of water, and as the water moved itself up the paper, and it dragged the inks with it. And that's exactly what's happening here. So the lady has peed on the end of the paper here, and then the urine is reacted with the antibodies if the hormone's present, and then it's gradually being moved up what is technically this chromatography paper, or something that's very, very similar. Now, what we have here is another type of, we have some more antibodies. Okay, and these ones have an enzyme attached to them. So I'm just going to put the enzyme here, and I'll talk about what that is in a minute. Now, there's different levels of things you have to remember for this. So if it's looking complicated, I'm going to break it down into simple, and I'm going to then take it up for the top end. So there's different levels that you can feel comfortable with as we go through. So bear with me with this. What's going to happen is we're going to end up with, effectively, let's put another colour on here. We're going to end up with effectively a hormone sandwich, so an HCG sandwich. Okay, because what's happened is this antibody here is binding here with the same antibodies. So we've got the antibody at the top, antibody at the bottom. So we've ended up with this sort of like a sandwich, and I can put in here sandwich. Okay, now if you remember, these hormones had a dye on them. So what we end up with is we end up with a reaction. And what that effectively means, it changes colour. Okay? So, we've peed on the strip. If the hormone's present, it binds itself to our antibody. This then gets pulled up the test strip to where there's some more antibodies, and we end up with this binding here. So, so we end up with this HCG sandwich, and we end up with a reaction between this enzyme, and I'll put enzyme there, and the dye, so we end up with a colour change. So that would indicate that the female is pregnant. Okay? Now, this is going to carry on moving up the paper, because what we need to find out, and this is the most important thing, is we need a control site. So what I will do in a minute, I'm going to draw the diagram out again, is if HCG wasn't pregnant. So we can talk through what would happen if the hormone wasn't there. Because what we need to know is actually, did this pregnancy test kit work? Because if it didn't change colour here, there's always going to be that moment of doubt, wonder if the test kit was faulty. So what they do is they put in a second window, and that should always change colour, whether the person is pregnant or not. And what this is, this will be a section where we've got the same antibody, but it's already bonded with the hormone. Which means, and it's still got the same enzyme on. There we go. which means there will always be some unreacted antibodies, which is going to bond. Okay, so this bit should always change colour, regardless of whether the hormone is present or not. So let me just show you what that would look like. So let me draw another line. And we'll draw the test kit out to start with, as it would look if you could sort of look in and look at the chemicals without actually putting anything else on there. So we're going to have some of our hormones in the control site. We're then going to have remember, this is all being done with our monoclonal antibodies. These ones are going to have the enzyme on them. Okay, and these ones are going to have the dye on them. And we said these ones. So this is effectively your test kit before it gets used. So we've got the reaction site, we've got the test site, we've got the control site. Lady's going to pee on the reaction site, but this time she's not pregnant. There's none of the HCG hormone present. Okay, so these antibodies are going to move their way up to here. But this time, there is no antibody present, so they cannot bind here. So if we just put down, cannot bind. So there's no reaction. Okay, they cannot make that HCG sandwich. So there's no reaction in any way, shape or form there. So that window stays blank. But obviously we need to make sure it's working. Now, because this one already has the HCG hormone there, we're always going to get a reaction. So this bit will still change colour. So there we go. Reaction equals colour change. Okay, so just have a think through what's going on, because this is quite complicated. But when you break it down, if you can understand this, you may in an exam be expected to label a diagram. They could ask you to break it down into simple steps. It could be a 6-mark question. But realistically, just bullet point. And even in a 6-mark question, if you don't want to draw a diagram, I would still be getting some scrap paper, and I'd probably be drawing something like this out so I can get my head around what's going on, because I find it really hard to explain these things without having that diagram. So don't be afraid in an exam to be able to do that, to sketch out what's going on, and either then annotate the diagram as part of your exam question, or then use the diagrams to produce some bullet points. Okay, so Tanishka, what's happening here, if you notice that the control site already has the HCG hormone bound to the antibodies. So regardless what's going to happen here, there will always be some of these will always bind to the control site. Yeah, these ones, we can only get a reaction happening if there's the hormone in the urine, so it combines here. But up here, there will always be a combination, it will always bind. So what we're looking at, P on the stick, if HCG is present, it binds to the antibodies. These then move up the site. If it's not present, the antibodies travel anyway. Okay, so we're going to get the direction of movement anyway. If it's present, we get binding at the test site and we get a colour change. If it's not present, there's no binding, so there's no reaction. It then moves its way up to the control site, and whether it's present or not, there will always be a binding at this section to prove that the pregnancy test kit works. Okay, so this is one of the uses that you need to get your head around. And I suggest spending a bit of time. If I haven't made it clear, look in the revision guide, go and have a look at some of the YouTube videos. What I was going to show you just briefly is if you, let me just bring this up here. If you just type in monoclonal antibodies into Google and then click on the images, there's lots and lots of other images that are showing you sort of similar things on here. So we've got there actually how it's produced. So we've got the production of them. We can scroll down and if we're not sure, let's just put in GCSE in there. That just helps filter it down a little bit more. There we go, we've got production here. Information about how pregnancy test kits work. And here again, okay? So there's quite a few different bits. We'll just bring that one up. There's quite a few different bits of information showing very similar to what I've just been talking about, but they may explain it in a different way that makes sense to you. So it's well worth going through, spending a bit of time, just get your head around that and make sure that you're happy with what's going on. Like I said, if you're not sure, just put it back up again. If you're not sure, please feel free to ask me in the Q&A session after this, so we can go back through, because I don't mind asking any questions, just to make sure you're really, really rock solid with it. Now, I'm not going to go through in that much detail for the other types of uses, but we just need to discuss, I suppose, how it works. Or sorry, not how it works, what the other uses are for this one. So, detecting pathogens is quite a useful way. So I said earlier that we could technically use this for testing for COVID-19. It's not as effective if there's lots of different variants of the pathogen, which often happens with the cold and with flu viruses. Because they mutate quite fast, the antigens will also change, which is why we can go down with a cold every single year, and it's not technically the same cold virus. Because it's mutated, the antigens are different, so our body has to learn to fight it every single time. And I think one of the problems with COVID is there's so many variants that we actually would need to have monoclonal antibodies produced for each variant, so it's quite hard to do. But if we had something more serious, or something that, let's just say, maybe it wasn't as common and there was only one type of that particular pathogen, then realistically we could produce those antibodies for the antigens, and we could put them on a test kit that changes colour in exactly the same way that the pregnancy test kit does. Then if we were to put some bodily fluids onto that test kit, if the pathogen was present, it would bind itself with those antibodies and we should get a reaction happening. So the same principle works for identifying whether a pathogen is present. Got to stop saying that. Other uses we can use it for, we could actually locate or tag particular tissue types. So for instance, if we were looking for a particular tissue type, deformity or some issue within the body, we could actually have it tagging up with a dye that may then show up under X-ray or maybe luminescent in some way. From a forensics point of view, if you think about if we want to try and have a dye that fluoresces, we could actually produce monoclonal antibodies with a fluorescing dye that binds itself to particular types of tissue types. So we could therefore then spray this around a room and we can actually use this to highlight areas where those tissue types, fluid types may be present. We're also looking at using this for tagging particular diseases. Now, I know that Southampton University has been working on, and not just Southampton, but other universities around the world as well. I just know a little bit more about Southampton Uni. They've been working on ways of tagging cancer cells with particular antibodies or antigens. Because if they do this, then actually all of a sudden the body's immune system recognises those cells as cells that shouldn't be there, and we can then allow our own immune system to start fighting some of those cells. Which means we then don't need to have toxic treatments such as chemotherapy, we don't need to have radiotherapy, and it's possible we may not need invasive treatments such as surgery. We're a long way off being able to do this for everybody, because actually every type of cancer is different and every body is different. So you would actually need to tailor each monoclonal antibody to that person's unique DNA and to their particular type of cancer, which means it's quite a slow process. And because cancer is quite big, we haven't got any techniques of doing this in a rapid way at the moment. But there have been some types of cancer, and if you do the research on this, where we have shown it to be quite promising, to use monoclonal antibodies as a way of tagging those particular cancer cells so that we can allow the body's immune system to fight it. Likewise, we could also use this for treatment like AIDS. Technically, if we can tag them, we can produce the antibodies, then we could actually get our body's immune system to have a bigger part in trying to fight some of those cells. So this is really huge in the world of science at the moment. And I think making sure the research still happens and goes ahead and understanding what's going on ahead is really important, because it could produce not just life-changing for small groups of people, but for large groups of the population, this could prove a life-altering situation. So make sure you're happy with that and how it works. Now, you don't need to know too many specific examples. The pregnancy test kit that we've just looked at can be applied to lots of different situations and scenarios. So don't be afraid if they show you a question in the exam and you go, well, I've not been taught that. Think about, can I apply what you know, what basically we've just done, to a different situation with that? Now, there are some disadvantages. We've obviously spoken about the mice, and there is an ethical issue. There always is when we come down to animal testing or animal uses, that it often causes the death of the mice. So we're causing that sort of destruction of life there. We've also got the issue that mice antibodies aren't as well tolerated by the human body. So we can get rejection, we can get reactions with the body, we can get some issues going on. But they're much, much easier to produce in mice than with most other organisms around there. So we continue to use mice because it's easy. We can produce large quantities very, very, very fast. But they're not as well tolerated by the body, so we get much higher levels of side effects with it. So it's worth being aware of that. We're looking at those advantages and we're looking at those disadvantages. So make sure that you could evaluate the use of monoclonal antibodies, particularly the ones that are produced from mice, and that you could talk about the advantages, the disadvantages, and then give your own opinion with a reason for that. Okay? What are we looking at? 22. Right, I think what I'll do, I'm going to talk about culturing of microorganisms and staying safe. This will be a recap from Biology 1, and then on Wednesday we'll do plant diseases, and then that'll finish off this topic. So we'll do that then. Okay, so let's just go through this now. I just need to see how far we would get with it. Right, so by culturing culturing cells and microorganisms, what we're looking at is growing them in a lab. So we can culture tissue types, we can culture human cells, animal cells. This can be used for drug development. Remember, we said that when we're looking at the preclinical trials, one of the things we will often do before we do anything else is we'll test chemicals on different cells to see if we get some form of reaction. So we need a way of growing them. On the other hand, if we are trying to identify a particular infection so we knew what antibiotics to use, we would have to grow those bacteria so we could look at them under a microscope, so we can actually try and identify what they are. It is possible to use monoclonal antibodies to have a test kit for different types of bacteria, which would be very, very fast. But that would only work if it was one of the common ones, because it's probably not possible to produce a test kit that would work on every type of every type of bacterial out there, because there's so many different types of antigens. But it could be used for some of the more common ones, like Staphylococcus aureus that we said is on our skin. So in other circumstances, we're going to have to grow and culture those cells so we've got enough of them that we can actually look at them under a microscope. And then even to the naked eye, when we get enough of them, you can actually start seeing them on a plate. So when I used to work in the food industry, so I used to work in the brewing industry and doing quality assurance, we would have to take swabs and product samples from lots of different points around the factory. What we would then do is we would grow them on a particular type of nutrient medium. So it was a type of jelly called agar, and there's different types of agar. And that's basically a jelly that has all the nutrients that bacteria like, so they grow very, very fast. We would then grow those in an incubator, so that's basically an oven that's at a temperature that bacteria like, which in industry is 37 degrees, same as our body temperature. In schools we only use 25, and we'll get these notes down in a moment. And then afterwards what we would do is usually after 3 days, we would take them out and we could just look at them, and any time there was a bacterial cell on there, they would have grown into a colony of bacteria, and there'd be enough bacterial cells that we would actually be able to see it with the naked eye. Now that's not the individual cells, but we could see the individual colonies. So I think I should be able to show you a picture of that. Again, we're just going to use Google for this one because it makes our life nice and easy. So if I just type in bacterial colony agar and then turn it on so you can see it, straight away, these are the sorts of things we were looking at. Every one of these dots would have been a bacterial cell, which has now grown into a colony. And remember, we said that bacterial cells multiply every 20 minutes. So actually, they're multiplying massively fast with this. So fast that we can see them with the naked eye. If we look at some of these, especially the furrier ones, will actually are actually types of fungi. And you can get these sort of identification grids, so we can actually have a look at which ones are bacterial, which ones are fungal when we look at them. But every single one of these would be a bacterial colony. Okay? And you can see they all look different. So straight away, that gives us an idea of what the type of bacteria is. It gives us an idea of some other type, basically what it is. And so we can start narrowing it down to smaller groups. We could then look at some of these under a microscope so we can get a closer idea to what they are. Some of these would also be grown what we call anaerobically. That's for next lesson, let's get rid of that, which is without oxygen, but we wouldn't ever do that in a school because often the nastier types grow without oxygen, but we would grow them in a particular cabinet. So let's just have a quick look then at how we would do this, and then we'll stop and we'll go on to Google Meet so that you can ask any questions that you might need. Now, when we look at, you would have seen a lot of these before. Generally what we have is some form of shallow plastic dish with a lid. These are called a Petri dish. And inside that, what we normally do is we'll put some form of jelly, and we just normally use a normal agar jelly. And I'll just put down, this is a nutrient medium. It's just a nutrient jelly. Okay. All of this would be sterilised. So everything I'm putting down here is going to be sterilised beforehand. So when we're making the jelly, normally what we do is we boil it. The Petri dishes either arrive sterile or we'll put them into some form of autoclave, which is a machine that heats up to high temperatures with lots of steam, because the steam gets into everywhere and makes sure that any bacteria are going to be killed. But a lot of the time they arrive pre-sterilised. We then put the agar into the jelly, we pour it out, and then the lids go on, and then it sets. So what we then do is we actually store these upside down. So if we were looking, let's just draw this out. Hang on, let's move a little bit more space on the desk. There we go. So we're going to store these upside down with the lid that way. We're going to put the jelly back in here. Okay, so we store them upside down, and the reason for this is this means that less likely. What am I doing? I can't even spell today. There we go. No. Wrong button, that one. So we've got a lower likelihood of microorganisms. That just means microorganisms dropping onto the agar. So remember, everything we're looking at is to try and avoid contamination. So we're looking at the key word here is aseptic. So we're avoiding contamination. Now, if we want to take a sample from an object, what we would do is we would get either a swab or a loop. So we often will have some form of handle with an inoculating loop, or we could have a swab. If it's one of these, we would hold into a flame to sterilise. And then we would take the base off the Petri dish, we would hold it up and we would wipe our sample onto the agar plate. Now, the other thing we used to do in industry is we would have these particular types of filter paper that we would pour some of our sample through, and then we would drop those onto the agar itself. So let me just see if I've got a picture of that I can show you. That's the antibiotic ones. I'm having a brain blank. I can't actually think what the discs were called. It looked a little bit like this. Okay, if you see this one here, we've got a disc that will just float on the top. I'll come back to these ones. These ones we actually spoke about in Biology 1. It's a different experiment. We could actually float it on top, and then we could actually count them on those discs. Okay, so that's the other method that we would have done. But everything we're doing is designed to stay sterile. Afterwards, what we do is we would write on the lid date. We would normally initial it, and then we would label what it is. Now, we do this on the base, not the lid. The main reason is if the lid ever falls off, we wouldn't have a clue what the sample is. And I have seen people walking along before with a tray of these, and they've tripped and they've dropped the whole lot on the floor when they've taken them out to sample them. At that point, it doesn't make any difference if there's contamination, because we're not waiting for anything new to grow because it's already grown. So if it's written on the base, we can just pick them up and read them. But if you write them on the lid and all the lids are separate, you've now wasted all of your samples. We store them upside down, and then we incubate. In a school, we would tape that way. Those of you that I've taught before, so I go on quite a lot about this. So if we've got a Petri dish, we tape that way. We don't tape around the outside, because what we don't want to do is seal off all of the oxygen. Because if we seal off the oxygen, we go into anaerobic, which means without oxygen, and usually the nastier pathogens grow anaerobically, and a lot of them will only produce the nasty toxins anaerobically as well. So from a school environment, we would never ever tape that way. We also incubate 25 degrees Celsius in schools, and we'd incubate a 37 to 40 in industry. That's because industries are better equipped at dealing with the pathogens. We don't want the high numbers produced in schools. We just want to be able to see that something there. So it's going to be much safer. So from our point of view. When we've finished, everything gets bagged, and then we put into an autoclave. And we cook it to kill off everything before we get rid of it. And it usually goes into biological waste. Okay, so that's just to make sure that nobody gets contaminated and nobody gets hurt. So main thing we're looking at culturing. We pour the agar in, everything stays sterile. We store them upside down. Then when we're ready, we take a sterilised inoculation loop, which we can dip into our sample. It's been sterilised by holding in a flame. We gently wipe it over the surface. We then stir it back upside down again. We write on the date, our initials, and we label it. We may want to tape it, but we only tape a little bit around the edges. We don't actually go all the way around to seal it. We don't want anaerobic. And then we incubate at 25 degrees for school, 37 degrees in industry, usually for about 3 days. When it's finished and we've looked at them, we've counted the samples, and we've done everything we need to do, we're going to bag it, we're going to cook it to kill off everything, and then we get to dispose of it in biological waste. Right, that is me done with this for today. So I'm going to finish that there. As always, I'm going to put the class code for Google Meet in the classroom in a minute. So give me 2 to 3 minutes. If it doesn't show up, refresh the page because it will be in there. And I'm going to be online probably till about 10 past 12 in case anyone has any questions. If you need me for longer than that, it's absolutely fine. But usually I'll just stay on there till 10 past 12 to see who turns up. Even if you've got questions, pop in and say hello. It's always good to see a few faces and make sure that everyone's doing well. Take care, guys, and I shall see you on Wednesday, if not in a minute.